Hello students, welcome back to Brilliant Education Doha, Qatar. In this video class, we are going to discuss one of the complicated topic from 11th grade, glycolysis. Glycolysis is that topic you would see in the plant respiration chapter, the different steps of glucose breakdown process. Glycolysis, Krebs cycle, electron transport system, electron transport chain, oxidative phosphorylation, various steps are there. The first step in the respiration is glycolysis. In this video class, we will discuss the all steps of glycolysis, end products of glycolysis, ATP tracking also. This video 100% helpful for you for the NEET like competitive purposes. So all of you carefully watch this video and write down the enzyme names and also stages of all this glycolysis process. So what exactly glycolysis process? We know this glycolysis process is also called the EMP pathway, Emden, of Parner. These three scientists explain this complete mechanism, glycolysis process. Glucose, one glucose molecule. So here one glucose molecule, glucose convert into this one glucose molecule convert into two molecule of pyruvate, and along with the two molecule of pyruvate we will also get the net ATP, net 2 ATP molecule and also 2 NADH also we will get. That means total at the end of the glycolysis 3 end products will form. What are they? 2 molecule of pyruvate, 2 ATP molecule, net gain I'm talking about the gross ATP molecule is 8 ATP, that we will discuss little later, and 2 NADH. You got my point? So total different steps, 10 different steps are there. Now we are going to start with the step by step. First step. Look at here. What exactly the first step of this glycolysis reaction? All enzymes also we will discuss now. Look at here. The first step. What is the first step? Glycolysis. Glucose. First, one molecule of glucose, 6 carbon compound glucose. This glucose takes one phosphate from ATP, and this glucose convert into glucose 6 phosphate. So here what I'm writing, this glucose takes one phosphate from ATP. So adenosine triphosphate. In adenosine triphosphate, 3 phosphates are there. The 1 phosphate is given to glucose. So glucose take 1 phosphate and that phosphate will be on the 6th carbon of glucose. So the glucose convert into the next step. Next compound is called what? Tell me. Glucose 6 phosphate. So the next compound is, I am writing here, it is glucose 6 phosphate. Am I clear? Glucose convert into glucose 6 phosphate. Now after taking off 1 phosphate from ATP, ATP convert into ADP, right? Now ATP convert into ADP. You got my point? So in this step, this total step is controlled by an enzyme called hexokinase. Hexokinase enzyme. Okay, this side I will write the all enzyme names also. So here in the step 1, glucose convert into glucose 6 phosphate after taking 1 phosphate from where? ATP. So that ATP convert into ADP, adenosine diphosphate. Next step, glucose. Now glucose 6 phosphate convert into its isomer called fructose 6 phosphate. Okay, this is a simple isomerase enzyme is participate. That means glucose convert into its isomer called fructose 6 phosphate. Glucose 6 phosphate convert into fructose 6 phosphate. So what is the enzyme name here? The enzyme name is isomerase. The enzyme name is isomerase. Now fructose 6 phosphate again taking 1 ATP, 1 phosphate from ATP. Now fructose 6 phosphate, this fructose 6 phosphate again taking one more, what tell me? Phosphate from ATP. Now look at here, ATP is giving one more phosphate and convert into ADP. Already glucose took one phosphate. Already in the first step, one phosphate is given, one ATP we already invested. Now this time second ATP, second phosphate is taking from the second ATP. You got my point? That means fructose 6-phosphate already having the phosphate on the 6th carbon. One more phosphate is taking. One more phosphate is taking from the ATP. Second phosphate will be on the first carbon of fructose. So what is the next compound now here? The next compound is fructose 1,6-bisphosphate. You got my point? Now fructose, this fructose 1,6-bisphosphate. Sir, what is this 1,6? Second ATP, that means second phosphate on the first carbon of fructose. First phosphate is on 6th carbon. You got my point? So how many total phosphates are there? 2. So biphosphate, fructose 1,6-biphosphate. So which enzyme is doing this action? Which enzyme is participating in this reaction? Write down the enzyme name. Fructokinase enzyme. Phosphofructokinase enzyme. This is phosphofructokinase enzyme. You got my point? So far, how many ATP we invested? 2 ATP we invested, right? Number 1, in first glucose to glucose 6-phosphate, 1 ATP, 1 phosphate come from the ATP, and fructose 6-phosphate to fructose 1,6-bisphosphate, 1 more ATP. In the NEET like competitive examination, they will give you at which stage ATP invested takes place during the glycolysis process, they will ask. So remember the 2 stages in which we invested the ATP. You got my point? Next, fructose 1,6-bisphosphate is now going to, this one is going to break down into 2,3 carbon compound. 2,3 carbon compound. Look at here. Sir, fructose 1,6-bisphosphate is how many carbon compound? 6 carbon. We know. Look at here. Glucose 6 carbon. Glucose here. Fructose 6 carbon. Fructose 1,6-bisphosphate 6 carbon compound with total 2 phosphates are there. 1 phosphate on first carbon, 1 phosphate on 6th carbon. Fructose 1,6-bisphosphate now divide into, that means it split into 3 carbon 2 compounds. Look at here. The first 3 carbon compound, I am writing here, it is called glyceraldehyde. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, 3 carbon compound with 1 phosphate. Next compound is dihydroxy acetone phosphate. Dihydroxy acetone phosphate. You got my point? This is also known as DHAP, dihydroxy acetone phosphate. Dihydroxy acetone phosphate also 3 carbon with 1 phosphate. So here it is. I am writing this is also 3 carbon compound. In presence of an enzyme called aldolase enzyme. Which enzyme? Aldolase enzyme. Now, student, keep in mind, from glucose to fructose 1,6-biphosphate is dividing into 2,3 carbon compound. Till here, step 1, that means phase 1 of glycolysis. So glycolysis completed in 2 phases. The first phase, from here to here, glucose to formation of what will make glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. Till here, this is phase 1, first phase. I am writing here, it is phase 1. I can write this is first phase of glycolysis. You got my point? First phase of glycolysis. Keep in mind, students, all of you, these 2,3 carbon compounds are isomers. That means they get interconvertible. That means Glyceraldehyde 3-phosphate convert into dihydroxyacetone phosphate. Dihydroxyacetone phosphate convert into glyceraldehyde 3-phosphate. Keep in mind. So which enzyme is participating here? Isomerase. Now which enzyme here it is? Isomerase enzyme. Till here everything clear, right? So this all steps clear, right? This is a step 1. That means phase 1. In phase 1 of glycolysis, 2 ATP so far we expended. That means so far 2 ATPs has been invested in this phase 1 of glycolysis. Am I clear? Now the next reactions, glyceraldehyde 3-phosphate from glyceraldehyde. Everyone keep in mind, I told you there are 2-3 carbon compounds are there. One is called glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. That means they both are isomers and interconvertible. Remember all of you, I am taking the rest of oxidation reactions from the glyceraldehyde 3-phosphate. Okay, and we have to double this one because dihydroxyacetone phosphate also convert into glyceraldehyde 3-phosphate. That's why keep in mind, whatever the reactions we are writing, that will be double. You got my point? Right. Now I'm taking glyceraldehyde 3-phosphate. Look at here what I'm taking now. I'm taking glyceraldehyde, this one, glyceraldehyde 3-phosphate. Steps. Glyceraldehyde 3-phosphate. 3-phosphate. How many carbon compound? 3 carbon compound. Already 1 phosphate is there. 3-phosphate. On the third carbon, 1 phosphate is there. Yes. Now look at here. This glyceraldehyde 3-phosphate takes 1 inorganic phosphate from PO4. So here what happened? 1 phosphate from PO4 it takes. That means 1 more phosphate is come and add. Right? Exactly this stage, NAD coenzyme, nicotinamide dinucleotide coenzyme, reduced to form NADH. You get my point? Exactly this step, what happened here? Exactly this step here, NAD convert into NADH plus H plus. Keep in mind, very, very important this one. 1 NADH equal to 3 ATP. Now this 1 NADH in electron transport chain will give 3 ATP. Remember, this is a check. This is not direct currency. ATP is direct currency. Now NADH is a check. We will encash this check into a ATP in electron transport chain. Okay, keep in mind. So far how many NADH has formed? 1. Which stage? Glyceraldehyde 3-phosphate is convert into glyceraldehyde 3-phosphate convert into the next compound. 1 more phosphate is taking from the PO4, right? So the next compound is 1,3-bisphosphoglycerate. Bisphosphoglycerate. Keep in mind. Why 1,3-bisphosphoglycerate? Already in glyceraldehyde 3-phosphate, 1 phosphate is there on the third carbon. 1 more phosphate is coming on the first carbon. So 1,3-bisphosphoglycerate. 2 phosphates are there. So 1,3-bisphosphoglycerate. You got my point? Now one more time I'm repeating this step. Glyceraldehyde 3-phosphate is 3 carbon compound. Let me write here. It is 3 carbon, 1 phosphate. 1 phosphate. That phosphate on which carbon? Third carbon. Phosphate on third carbon. Again it take phosphate from PO4, inorganic phosphate. That inorganic phosphate comes on the first carbon. So glyceraldehyde 3-phosphate convert into 1,3-biphosphoglycerate. Now tell me student, 1,3-biphosphoglycerate have how many phosphates? 2 phosphates, right? One is on first carbon, one is on third carbon. That's why we are calling what? 1,3-biphospho or bisphosphoglycerate. You got my point? The enzyme. The enzyme involved in this reaction is, what is the enzyme name? Glyceraldehyde 3-phosphate hydrogenase. Hydrogenase. hydrogenase enzyme. Glyceraldehyde 3-phosphate hydrogenase enzyme is that means catalyze this enzyme. That means this enzyme will convert this glyceraldehyde 3-phosphate into 1,3-biphospho. That means 1,3-bisphosphoglycerate. Now 1,3-bisphosphoglycerate have 2 phosphates. 1 phosphate on first carbon, 1 phosphate on third carbon. Yes, students? Look at here. Now all of you look at here. What happened? This 1,3-bisphosphoglycerate give 1 phosphate to ADP. Remember, all of you look at here. Now what happened? 1 phosphate it is giving to ADP, diphosphate, adenosine diphosphate, taking phosphate from the 1,3-bisphosphoglycerate, and now it convert into ATP. My dear student, this is the first step in which ATP synthesis takes place in glycolysis. You got my point? 1,3-bisphosphoglycerate have 2 phosphate. One is on first carbon, one is on third carbon. 1 phosphate it's given to ADP, so that ADP convert into ATP. Question: in 1,3-biphosphoglycerate, how many phosphates left? How many left? 1, right? You got my point? That phosphate is on the third carbon. That means this 3-phosphoglycerate will form. So what is the next compound name? Next compound name is 3-phosphoglycerate. Glycerate. You got my point? Sir, why did you remove that bisphospho? That means 2 phosphates is there. We use the bisphosphate. Now it is 2 phosphates are not there. So I removed the bisphosphate. So simply I am calling what? 3-phosphoglycerate. Question: 3-phosphoglycerate have how many phosphates? 1 phosphate. On which carbon? Third carbon. So we call what? 3-phosphoglycerate. You understand? Remember, all of you, in NEET like competitive purpose, they will ask at which step ATP synthesis takes place in glycolysis. Everyone note this point. 1,3-bisphosphoglycerate convert into 3-phosphoglycerate. Exactly this stage ATP synthesis takes place. You got my point? And this reaction controlled by an enzyme. The enzyme name is phosphoglycerate kinase. Here we can write phosphoglycerate kinase enzyme. Phosphoglycerate kinase enzyme. Next, 3-phosphoglycerate, phosphate on third carbon, just it shift the phosphate position from third carbon to second carbon. Nothing changed. Just what happened? Phosphate on the third carbon. So phosphate will shift from the third carbon to second carbon. So that the next compound is 2-phosphoglycerate. So the next compound we can write here, it is 2-phosphoglycerate. 2-phosphoglycerate. You got my point? So here what happened? 3-phosphoglycerate, that means phosphate on the third carbon, just shift the number of carbon. That means phosphate from the third carbon, it go to second carbon. So 2-phosphoglycerate. That means 2-phosphoglycerate still have 1 phosphate, student, keep in mind. You got my point? Now here, it is phosphoglycerate mutase enzyme. Which enzyme here? Which enzyme will catalyze this one? Phosphoglycerate mutase enzyme. Now 2-phosphoglycerate convert into phosphoenol pyruvate. So now this 2-phosphate convert into phosphoenol pyruvate. Now this here also one enzyme is there. Which enzyme here? This enzyme is known as enolase enzyme. These all are enzymes. Enolase enzyme. Sir, I have a question. Phosphoenol pyruvate have how many phosphates? 1 phosphate, right? Yes or no? 3 phosphoglycerate, 1 phosphate. Just phosphate shift from third carbon to second carbon. 2 phosphoglycerate, 1 phosphate is there. Now here phosphoenol pyruvate also having 1 phosphate. That last phosphate is given to ADP to form ATP in the next step. Now what happened? This phosphoenol pyruvate finally convert into pyruvic acid or pyruvate. Pyruvate. You got my point? The enzyme name is, here the enzyme participate, the enzyme name is phosphokinase pyruvate enzyme. Phosphokinase pyruvate enzyme. So now keep in mind exactly this place what happened. Now last phosphate is given to ADP. So ADP is taking last phosphate from phosphoenol pyruvate to form ATP, second ATP formation. My dear student, look at here. 2 places so far ATP has formed. Number 1, exactly this place. ATP has formed. First ATP 1,3-bis phosphoglycerate form, convert into 3-phosphoglycerate. This step first ATP has formed. Second ATP has formed between phosphoenol pyruvate to pyruvic acid formation. Second ATP has formed. You got my point? Now tell me, wait a moment. Here we took glyceraldehyde 3-phosphate. Now glyceraldehyde 3-phosphate, further all step. This total steps are known as second phase of glycolysis. So the second phase of glycolysis is energy releasing phase, student. First phase energy investmented phase, second phase energy releasing phase. Keep in mind how many ATPs has formed here. 2 ATPs has formed. Wait a moment, one more important thing keep in mind. Glyceraldehyde 3-phosphate has participate in this all reactions to form what? 2 ATP 1 NADH. Yes, let me write here 2 ATP 1 NADH. What about this one? Dihydroxyacetone phosphate also convert into glyceraldehyde 3-phosphate, and this reaction will be repeat one more time, right? I told you 2 times we have to double these reactions. Now tell me, into 2, that means we can write total 4 ATP formation plus 2 NADH formation. But my dear student, 2 ATP we already invested in the first phase, right? So out of the 4 ATP which we form here, which we get here, give 2 back. So 4 ATP minus 2 ATP. This 2 ATP I am giving here. We already invested here. So I am giving back. So how much of a net gain of ATP? 2 ATP. 2 ATPs are net gain. So here I am writing net gain in glycolysis. In glycolysis, 2 ATP net gain, remember. So what about the NADH? Now how many NADH? 2 NADH, right? So here 1 NADH is equal to 1 NADH give 3 ATP. That means 2 NADH into 3 ATP. That means total 6 ATP. But this 6 ATP will not form in glycolysis. We have to encash this NADH2 into ATP in where? Electron transport chain. That means the gross ATP, the gross gain of ATP in the total glycolysis process, 6 plus this 2 ATP, 8 ATP. 8 ATP is what? Gross gain. You got my point? Net gain of ATP, 2 ATPs direct cash, that means direct ATPs, but here we need to keep in mind 2 NADH convert into ATP later, that will give 6 ATPs. You got my point? So in this total glycolysis steps, in this total glycolysis process, look at here. Step number 1, 1, 2, 3, 4, 5. First 5 steps are phase 1 glycolysis. This is energy investmented phase. Now the last step, that means the rest of steps are what? Tell me. Total second steps are energy releasing phases. Energy releasing phases. So all of you keep in mind, in energy releasing phases are the second phase, energy investment phases are first phase. Clear? Everyone keep in mind. One more time I am repeating. At the end of the glycolysis, 2 molecule of pyruvate. Look at here. From one glyceroldehyde 3-phosphate, 1 pyruvate. From 1 dihydroxyacetone phosphate converted into glyceroldehyde 3-phosphate, 1 more phosphate. That means at the end of glycolysis, 2 pyruvate. At the end of the glycolysis, 2 pyruvate. Next, number 2. How many ATPs now? 1 and 2. There are 2 ATPs. That means at the end of the glycolysis, 2 ATP. 2 ATP. How many NADH? 2 NADH. These are the end products of glycolysis. Keep in mind, this step is very, very important. Many times question I've been asking, what are the end product of glycolysis? You got my point? 2 minutes. Let me revise this total concept one more time. Glucose take 1 ATP from glucose take 1 phosphate from ATP to form glucose 6-phosphate. Where is the phosphate? Phosphate on the 6th carbon. Now glucose 6-phosphate convert its isomer called what? Fructose 6-phosphate. In the enzyme name is isomerase enzyme. Fructose 6-phosphate convert into fructose 1,6-bisphosphate after taking 1 more ATP, 1 more phosphate from ATP. That means 2 ATP invested. Step number 1, ATP invested. Step number 2, ATP invested. Fructose 1,6-bisphosphate is breakdown into 3 carbon 2 compounds. Glyceroldehyde 3-phosphate, dihydroxyacetone phosphate. These 2 components are 3 carbon components. These are isomers. They are interconvertible. They are interconvertible. So I am taking what? Glyceroldehyde 3-phosphate. Look at it, the glyceroldehyde 3-phosphate. Glyceroldehyde 3-phosphate has 1 phosphate, 3 carbon compound. On the 3rd carbon, phosphate is there. Glyceroldehyde 3-phosphate taking 1 more phosphate from where? This time PO4, not from ATP, PO4 it's taking phosphate. Now it convert into 1,3-biphosphoglycerate. The enzyme is glyceroldehyde 3-phosphate hydrogenase enzyme. Next, exactly this step coenzyme NAD coenzyme reduced to form NADH plus H plus. 1 NADH is worth of 3 ATP. It will give in electron transport chain. 1,3-bisphosphoglycerate has now 2 phosphate, 1 phosphate on 1st carbon, 1 phosphate on 3rd carbon. Are you with me? Now 1,3-biphosphoglycerate give 1 phosphate to ADP, so that ADP convert into ATP. So the first step in glycolysis, ATP formation takes place. You got my point? Next, 1,3-biphosphoglycerate after giving phosphate to the ADP, it form ATP. So it's convert into 3-phosphoglycerate. 3-phosphoglycerate has how many phosphates? 1 phosphate on 3rd carbon. 3-phosphoglycerate is convert into 2-phosphoglycerate. That means the phosphate position will change from 3rd carbon to 2nd carbon. Next, this 2-phosphoglycerate is convert into phosphoenol pyruvate, and last phosphoenol pyruvate, whatever the last phosphate is there, it is given to ADP. So the ADP convert into ATP. Second ATP formation in glycolysis. So phosphoenol pyruvate after giving phosphate to ADP, it convert into pyruvate. So at the end of the glycolysis, 2 molecule of pyruvate, 2 net gain of ATP, 2 NADH formation takes place. Very important steps these all are, my dear students. So all of you go through these steps and ATP tracking, NADH2 tracking, and also what? Pyruvate end products. Okay, this video sure will helpful you to get a good marks in the NEET like a competitive purpose. So all of you, this glycolysis video, once again go through this one, all steps. No need to confuse. It's very easy to track the all 10 steps. Okay, thanks for watching.
Brilliant, Qatar. your trusted coaching partner for IIT, JEE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.